Welcome to this exciting session on advanced restoration through research and innovation, where we explore the groundbreaking KSA native plant genome pilot project. This initiative is a collaboration between the National Center of Vegetation Cover Development and Combating Desertification and King Abdullah University of Science and Technology. This collaboration is a beacon of scientific innovation, addressing critical challenges like biodiversity loss and land degradation while aligning with Saudi Arabia Vision 2030 and the Saudi Green Initiative. Before we begin, I would like to extend our heartfelt thanks to the G Global Land Initiative for providing us with this platform to share our progress and insights. Your support had given us the time and space to spotlight these critical efforts and ecosystem restoration and demonstrate how they contribute to the global priorities. Let me take a moment to introduce myself. My name is Mudawi Al-Ohali, and I'm a proud member of a research and innovation team at NCVC. I am both honored and privileged to be part of this institution. That is forefront and addressing some of the pressing environmental challenges of our time. Joining this remarkable organization has been humbling and rewarding journey. I am continually inspired by the dedication and innovation of my colleagues. So we are honored to have esteemed speakers. First, I would like to introduce Professor Ahmed Al-Ghamdi. Professor Ahmed Al-Ghamdi is the head of the Research and Innovation Department at NCVC in Saudi Arabia. With a PhD in plant ecophysiology from University of East Anglia, UK, he's an expert in plant ecology, biodiversity, and conservation. Formerly professor at the University of Hayel, Dr. Al-Ghamdi had authored numerous publications and books and contributed to sustainable development initiatives in arid regions. His work focuses on advancing environmental sustainability and combating desertification. Okay. Next, I would like to welcome Dr. Zhao Xiu, Professor Zhao Xiu. He's the Deputy Director of Ecology Department at the Institute of Cold and Arid Region Environment and Engineering, Chinese Academy of Science, and Director of Neiman Station. Since 2006, he has served as a director of East Asia Landscape Ecology Society and vice chairman of the Chinese Soil and Water Conservation Society. Received the first prize of scientific and technological progress in Gansu province and participated in research and ecological project on desertification in Inner Mongolia, China. Next, I would like to introduce Professor Rod Wing. Professor Rod is a scientist specialized in plant genetics, genomics, and evolutionary biology crops and their wide relatives. He serves as a professor of plant science and previous director of the Center of Desert Agriculture at KAUST. He is also the director of Arizona Genomic Institute. His research focus on developing sustainable, climate-resistant crop, particularly rice. With over 30 years of experience, Professor Wing had esteemed academic position, contributed to groundbreaking genomic projects, and lead initiatives like the KSA Native Genome Project. His work aimed to enhance desert agriculture and ecologically sustainability. He earned his PhD in genetics from USC Davis and conducted postdoctoral research at Cornell and UC Berkeley. I also would like to introduce my colleague, Dr. Asil Sentley, from our NCVC team. Dr. Asil Sentley is a senior research scientist at the National Center of Vegetation Development and Combating Desertification, specialized in bioinformatics, genomics, and biodiversity. With a PhD in bioscience from KAUST, Dr. Asil has led environmental research, contributed to Vegetative Cover Initiative and authored impactful publications. Her work focuses on sustainability and advanced environmental strategies. Also, I would like to welcome Dr. Nahid Muhammad from Dr. Wing's team. Dr. Nahid Muhammad is a research scientist at KAUST and part of the Center of Desert Agriculture, specializing in plant genome studies. With MS in molecular biology from University of Birmingham, PhD in biology from University of York, and her work focused on sequencing plant genomes, including culturally and ecologically significant species of Saudi Arabia. Dr. Nahid led the Native Genomic Project with a collaboration with NCVC, aiming to enhance biodiversity conservation, sustainable agriculture, and food security in the kingdom. Also, I'd like to introduce Dr. Majid Saad. He's from Professor Hertz's team. Dr. Majid Saad is a senior research scientist at KAUST Center of Desert of Agriculture and coordinator of Darwin 21 initiative, focusing on sustainable agriculture in arid regions. He holds a PhD in microbial genetics and has extensive experience in plant virology and microbial solutions for improving soil health and crop productivity. Dr. Saad also is the CEO of GrowBioM, an agri-biotech company that advances in sustainable agriculture practices. And lastly, I would like to introduce Dr. Muhammad Salim. Dr. Muhammad Salim is from Professor Salim Al-Babli team. Dr. Muhammad Salim is a research scientist at the Center of Desert Agriculture at KAUST. His expertise lies in metabolomics, focusing on large-scaling analysis and metabolites with complex biological systems. His research aimed to explain the rule of root and root exudate metabolites in plant rhizosphere communication. Dr. Salim specialized in metabolite profiling, fruit chemistry, and development of LMS analytical method and isolation of natural products. Together, they will guide us through the project vision, methodology, and global implication. This project exemplifies how cutting-edge science, spinning genomics, metabolomics, microbiome research, and root system agriculture can drive sustainable restoration and arid ecosystem. We'll explore not only scientific breakthrough, but also the possible application for biodiversity, sustainable greening, and ecosystem resilience. And now I would like to leave you with Dr. Ahmad's welcoming speech.
بسم الله الرحمن الرحيم السلام عليكم ورحمة الله وبركاته. Thanks Madawi for this really fantastic introduction. MashaAllah, we discovered a talent, a new talent in our department. So more than welcome. We already introduced you to the audience. Okay, so I'd like, in fact, to start this presentation, which is Advancing Restoration through Research and Innovation, the KSA project model. Okay, I think I can much better. The why research-driven restoration. Thank you. All of you know that biodiversity loss and degradation threaten ecosystem resilience and food security. This is now the talk that which taking place in this COP. Land degradation in general and desertification, which leads to biodiversity loss, which leads to many threats, among of them the food security. So that's why our project, which is a collaboration between us, between the National Center for Vegetation Cover and Combating Desertification and Abdullah University for Science and Technology, KAUST. And here is our respected colleagues from KAUST. So this project will focuses on genomics, microbiome analysis, and root architecture, and and microbe analysis as microbiome analysis as well, to address these challenges, also to fulfill the goals of the Saudi Green Initiative and the Saudi Vision 2030. But before we start the project, we'd like to give a general overview about the ecosystem in Saudi Arabia and the different vegetation cover in Saudi Arabia regarding these. One could imagine, especially people from outside the kingdom, that Saudi Arabia all deserts with few plants, with camel and maybe date palm and some acacias. While in fact we have rich flora in Saudi Arabia. We have about 2,300 species all over the kingdom. This is because of the Saudi Arabia is a vast, huge country, consists of many terrains with different ecosystems. For example, we have the vast area of the kingdom is, we call it rangeland. Rangeland means these areas with little trees and more shrubs and grasses. So 70% of the country are rangeland, and you will find over there many of these range species, including acacia, leptidina, Lycium shawai, and Salsola, Atriplex, Rhotorium, etc. So this is one ecosystem over there in Saudi Arabia, more than 70% of the country rangeland. Then when you go to the sand dunes, we have also three sand dunes in Saudi Arabia. The biggest one is the empty equator, which is the Rub al Khali, and the Dahna, and the Nufud al Kabir. And also, even those are, as I told you, sand dunes and very harsh environment. However, they still resembling natural ecosystems, having many species, among them species. And for example, for the plant species, we have al-ghada, haloxylon, and al-arta, caligonum, and Procybitinaria, اللي هو الغاف الرمادي. When it comes to the southwest of Saudi Arabia, it is the most rich area in terms of biodiversity, because it lies on the southwest highlands of Saudi Arabia. It receives a lot of rain, and the temperature is really nice most of the year. That's why we have forests over there. We have juniper forests. We have other trees. Most of the biodiversity in terms of plant species and vegetation lies in this area in the southwest of Saudi Arabia. When it comes also to the rest of the mountains towards the north of Saudi Arabia, which align with this the Red Sea coast, also we have Jibal al-Hijaz mountains, and also this mountains hosts a very rich diverse of species. For example, wild olives, wild figs, and also wild almonds. We have wild almonds in these mountains. Also, we have, as you see, the long seashore in the Red Sea in the west of the country, and in the Gulf Sea in the east of the country. These shores also, we have over there the coastal forests, which consist mainly of mangroves. So we have mangrove in the Red Sea coast and mangrove as well in the Arabian Gulf. So this is like an overview about the vegetation in Saudi Arabia. As I told you, we have more than 2,300 species. When it comes to trees, which is these species that have a trunk longer than 5 meters, we have more than 103 species. When you speak about shrubs, we have more than 340 shrubs here in Saudi Arabia, native shrubs, local shrubs. And we have more than 1,600 herbs and grasses in Saudi Arabia. Again, as I told you, we have 70% of the country rangeland, 1.2% of the kingdom's area are forests, and also we have almost 200 square kilometers of mangroves along the two coasts. So here is Dr. Salih Al Zamanan. He's looking I don't know about this figure. We'll check it with you, inshallah. Almost. We took it from FAO. You can speak with the FAO. So uh, so again, when we speak about the biodiversity challenges, as we told you, our country suffers very similar to other countries from biodiversity loss because of many factors. One of them is the land degradation because of anthropogenics and the climate factors. So really, more than 70% of the Saudi area, the Saudi country area, suffers from degradation. In order to restore this area, we collaborate with our esteemed colleagues from KAUST to do this program, or let's say this project, which is we call it the native genome, the native plant genome project, in order to analyze the genomics, also to analyze. Come on. Also to analyze the micro metabolomics in order to understand plant biochemistry and adaptive traits. Also to analyze the microbiome to try to elaborate more in the interactions between microbes and the ecosystem, and the root architecture and the seed germination. This is one also of the variables that we are working in this project. And in order to build our genomic database and biochemical resources in order to support the reforestation efforts by NCVC, in order, inshallah, to green our deserts and to train our Saudi scientists, young scientists. And we are really lucky today to have one of those scientists. Where is Asil? She is here. Asil Santri, she is one graduate of KAUST and one graduate of this program. So we are really happy to have this sort of outcomes of this program. And all this program is about a pilot with KAUST. And inshallah, we are having a new project, a new program will shed light also in more and more native species here in Saudi Arabia. And now I leave the floors to Prof. Rodwing from KAUST. Thank you, Rod.
Thank you. Thank you, Ahmed. Okay, thank you very much, Dr. Ahmed, for introducing the project. My name's Rod Wing. I'm a professor at KAUST, and it's been a real pleasure, honor to be able to work in Saudi Arabia and to work with a number of really great people here in the country. So I'm going to give you a little bit of an overview on the genomics and the vouchering side. So what kind of our idea around the project is that we identify what's called a voucher specimen. And that voucher is something we find in the wild, and then we tag it, and then we isolate DNA from the leaves, and we study the microbiome, the plant genome, the metabolome, the root architecture, the soil atlas, and so forth. So that this is kind of a ecosystem. So we're basically making a reference ecosystem, and then the idea behind that is once we have that, then we can use that information to assess the genetic diversity, the just the diversity of these species across the country. So we're starting out with basically a reference set, and then using that to attack the problems from a variety of avenues. So we received funding to start a pilot project. The pilot was to characterize 15 terrestrial ecosystems. So we started out with the vouchering right here. These are some of the locations. And then we did the genomics and the microbiomes. This is the Wing and Hurt labs. Herbert Hurt, Magid Saad is representing the Hurt lab there. The metabolic profiling, which is Salim Al-Bibi's lab, and Mohammed Saleh is representing that work there. and then the root development and architecture from the Ikram Blaloo's lab. And she couldn't make it to this meeting today. And finally, data release. The plan is to release all of the sequence data to the National Center for Biological Information. Okay, so this is we have a pilot right here, and this is just some examples of some of our activities. And the fantastic thing about this project was I've been able to see the whole country, and with many of these people here, including Ahmed Al Ghamdi. So really sampled the plants and shared bread and talked about science and very exciting. So I'm very fortunate to be able to do this. So this is our pilot ecosystem study focused on species critical for biodiversity and cultural significance, desert restoration, biodiversity enhancement, cultural and ecological importance. Genomics, genetic studies help promote afforestation, reforestation, and desert restoration in Saudi. And some species studies in the project are suitable for reforestation programs, while others are cultivated around desert plantations and windbreaks. This is the list of the 15 species right here. Here's a few samples here, and the results inform ecosystem-specific restoration strategies. Okay, so I want to just give two examples of some of the plants we've studied so far. The one is related to the Fast Fit Palms project. This is when I came here, we didn't have a very good reference genome for date palm. We now have over 20 reference genomes for date palm. And the first one and the best one that we have is the Ajwa date palm. We went to Medina and identified a voucher plant and have done all of the experiments, metabolics, microbiomics, and root architecture, leaf architecture, and the genome of the Ajwa date palm. So this is the highest quality genome out there, and we're working on a manuscript right now to publish this. Another example, and this is one of my most favorite examples of applications. This is everybody in the audience, probably most of you know what this is. This is Meswak, or a rock, and the scientific name is Salvador persica. In Muslim culture, they know this as they call this a rock or Meswak, but I call it the Saudi toothbrush. And I hope that's okay. I hope I don't offend anybody calling it Saudi toothbrush. But it has an extensive root system, and the roots are harvested and peeled back and using essentially to clean your teeth. So that's very exciting. But this is a magical plant in that the extensive root system anchors the desert floor. You can water it with salt water. But another amazing piece is that it produces berries that they look like blueberries. And this is something that is edible, highly nutritious. And one thing we're trying to do is hopefully try to neo-domesticate this plant. So make it so that you can actually grow this plant in the desert, water with salt water, and produce millions of berries that could be purchased at the grocery store. Rather than importing berries from Ecuador and Peru, like we're doing now in the Tamimi market, we're actually producing them locally in Saudi Arabia, but at the same time combating desertification. So this is kind of a model plant that we would really want to push. But it takes you can't just do this overnight. You have to do all the basic research to figure out how to do this. I think with that. Okay, so here's some of the genomics highlights. We isolate high molecular weight DNA from the genomes. This is done this has been done by Nahed Mohammed, and I can't believe your name. I forgot your name, Nahed, and the face of the project. And so these are the projects we have very, very high quality reference genomes. and we use and then this is the we also take herbarium specimens. So you take samples, make herbarium specimens, and also are preserving the seed. And this will all go into a seed bank, and then that's being led by our group, and then the root system architecture is led by Ikram Ballou. So learning about how the roots grow and develop in extreme environments. I think with that, the next slide. Okay. Okay, so now I'm going to hand this over to Mohammed Salim, who's going to talk to you a little bit about the metabolomics highlights. Mohammed?
Thank you, Rod, for your introduction, and good afternoon, everyone. It's my great pleasure to share with you some slides related to the metabolomics part of this native genome project. So my name is Mohammed Salim, and I am a research scientist representing the Salim Al-Babili lab. So we are specialized on plant metabolite profiling, targeted and untargeted analysis, discovery of plant biochemistry pathways, and discovery of the importance of bioactive metabolites, like in medicinal application as well as in agricultural applications. So our idea here is to do profiling of all these native plants from their environment. So we focused on different uh, organs, like we take samples from leaves, we take samples from roots, and we do analysis of all metabolites, primary and secondary metabolites. We have established workflow where we collect the samples, dry samples and fresh samples. We directly extract them, and we use different types of platforms like liquid chromatography and gas chromatography coupled to mass spectrometry for analyzing these samples. After we got the samples, we identify the major compounds, and then we assign these compounds to their biochemical pathways and to see the difference between these metabolites. So we have a workflow for the mass spectrometry-based metabolomics. We analyze the samples in different modes, and then we filter the samples for only important metabolites that might be promising for the biochemical pathway and for the bioactivities later. And then we do the compound assembly, and after this we do statistical analysis based on these results. So here I show you an example of the types of metabolites that we could identify. Actually, we could identify over 400 different metabolites from all the samples belonging to different metabolite classes. As you may see from here, we have metabolites from amino acids, we have from peptides, we are interested in sugars and this stuff, but we are also interested in different types of specialized metabolites that are markers for every single plant, like phenolic compounds representing biphenolic acid flavonoids, for example, or other compounds such as those found in Salvadora, the isocyanates, or different types of sulfur compounds that also provide medicinal and agricultural applications. So after we analyze these samples, identify different metabolites. Here on the left side, for example, you can see a principal component analysis where you can see how the samples are clustered together based on their chemical pattern or the different biochemistry background. You will nicely, for example, see here how samples like this is haloxylon, this is salsola, they are from the same family, Amarantaceae. They are clustered together based on that they have more or less similar metabolite profile. Samples from different ecosystem, like different plant family or different conditions, they will be clustered. And on the right side, you see I just show an example of the heat map showing red for increase of these metabolites and blue for decrease. And then among every single plant, you can see how specific metabolite classes could represent a specific plant ecosystem. For example, if you will look for the medicinal value of Salvadora persica, you will assign the value of these plants to, for example, a chemical class that's called sulfur compound isocyanate. And if you look at the heat map, you will directly pick that Salvadora persica here is shown, for example. And this is the highest red in glucosinolates, which is the sulfur containing compound. So we analyze every single metabolites from this sample, and we have 3 different targets. One of them to assign these metabolites to their biochemical pathway, and then by doing this, we will understand how these plants are adapting to this harsh condition in KSA, and then you can learn from this to transfer the model to other crops and species. And the second, to discover some metabolites that could allow plants to resist types of abiotic stress, something like parasitic weeds, something like nematodes, how these plants are handling with this based on their chemical profile. And on the third, we are trying to pick up which specialized metabolites can be used for medicinal application, something like the berries or berries like fruits, kabath from Salvadora, what's the nutritional value, how this can be implemented in different types of formula and products. Thank you very much. So I will hand over to my colleague Majid Saad for microbiome.
Okay, yeah. Assalamu alaikum. So I'm Magid Saad. I'm a researcher at KAUST, and it's really my pleasure to be a part of this great project. And I introduce within our lab a new concept is a metabolome or microbiome. Sorry, not metabolome. And then I wanted to just highlight here in the left panel that microbiome is really inhibited in every part of our human body. And it's exactly the same what is happening in plant. So the plant already are living, it's not only the genomic of the plant, but also there's another microbiome are living in everywhere in the plant. Outside of the plant or also inside the plant, from the shoot system, from the root system. And they are playing a very important function to help the plant to tolerate stresses, also to cope with different environmental changes, and helping the plant to acquire nutrient from the soil as well as the defense mechanism. And we introduce this concept of that the plant microbial and soil interaction in the genomic concept, that we not only need to know the genomics or the metabolomes which is being secreted by the plant, but also the interaction between the plant within an ecosystem, where the root system are interacting with the soil, and there's a feedback as well. What is the microbiome are playing the role between the connection between the plant system and the soil system? And here we already established a platform for screening or identifying all the microbial community from different ecosystem with the soil which been collecting the native plant. Along with the genomic sequence was being done with the Wing Lab, we did the microbiome sequencing in several levels, the level of bacteria, or what is inside the plant, and fungi as well. Not only in the plant as a leaf system, but also in the soil, because it's also feedback that the microbe between the soil is going to be in the plant again and go back again. And then we already have a 3A, who is there and what they are doing function, and how can we use this knowledge combined with the genomic sequence to help in the Green Saudi initiative and go forward to be more sustained in planting the tree. What is the major issue? It is water. So can we use this microbial community to decrease the amount of water used by the plant for the plantation? Second, the stability and the resistance. We know that sometimes that we have an issue with a lot of waste, which is mentioned about this metabolomic product which been secreted. Can this microbial community can help the plant to increase? And this actually comes with a third point: how can we translate this work, which is something which is very important from the Native Genome Project? What is the outcome, which is then the people can use it, not only for planting new trees, but also for continuing this 10 billion tree for the Saudi Green Initiative? That by understanding the microbial community in the root system, in the soil system, we come with a new concept of having inoculum, which have this bacteria has been identified, know the function of this bacteria, and preparing in a larger scale. And it will become like a kit when you are putting the new seeds for the Saudi Green Initiative. You have only the bacteria, you know the best genotype to plant, the best soil you can use, as well as the microbial community, which is can go together. As a result of what we have now, we already use the 15 plant from the Native Genome Project, and here's the profile of microbial community. We use a marker gene, which what we call it 16S, and we could see the high diversity of microbial community. A common microbial community is already present in every plant. However, we have some special microbial community are appearing in different plants. And here, for us, we are not looking to say who is there, not only, but what they are doing. Because there is a lot of microbes, they can have a redundancy on their function. Maybe they are different group of microbial. And what's important for us, what they can do. And you know the challenge in case A, heat, drought, salinity as abiotic stresses and biotic stresses. So we analyze this microbial community from a function, and we know what is the group of the function are connected with different microbe with which you plant. As we guys mentioned about the Salvadora, we have a high number of bacillus strains and the actinobacteria, which is a really highly producing an antimicrobial effect component, which is go very well with the use of this SWAC or the Salvadora for as an anti for a brush tooth or the killing the microbe. So with microbial, we are trying to close the gap. What is the microbial community, which is related to the genotype of the plant, as well as what is the metabolomic can be secreted. Because most of the metabolomic was been shown before might be specific for the plant, but also might be the microbe are one of the components which are able to secrete the component as well. So this is our new concept that we are not only at the genomics, not only a metabolomic, but at an approach of combining the genotype of the plant, the genome sequence, the metabolomic, the soil microbial community, and soil chemical and physical approach, and find the best microbial community. And by this one, we can have changing the non-optimized soil for the plantation to a healthy soil by helping the microbial community. And here I would like to stop and thank you very much.
Well, as it was highlighted, the collaboration between NCVC and KAUST was very important, not only in the native plant genome project, but also we established a collaboration in creating an NCVC KAUST experiment station and ecological observatory in Wadi Gdeid National Park that helps both entities advance environmental research and initiatives and support Vision 2030 and the Saudi Green Initiative. That station features a secured 10-hectare site with nurseries, seed banks, greenhouses, labs, and visitor facilities that have completed the first stage in October 2024. The core goals of this station include conducting studies on plant genomics, adaptation, and desert restoration, as well as implementing cutting-edge technologies like hydroponics and fog harvesting. And lastly but not least, to develop sustainable methods for desert agriculture and deforestation. There are many or various targeted research that can be implemented in this research station. It includes genetic studies on drought-resistant plants, as was like what was already mentioned by Dr. Majid and Professor Rod, Salvadora Presca. Integration of renewable energy with desert farming, as well as advancing water management solutions such as mobile desalination systems. Those two contributions and collaborations contribute in a local relevance and a global relevance. The global relevance, as we can see highlighted in the slides, showcase the contribution to the UN Decade and Ecosystem Restoration and G20 environmental priorities. Applications for resource restoration is similar in similar arid environments around the world can be applied here in the kingdom. As well as it aligns with the SDG 13, 15, and 17 are in the red boxes. In Life on Land is 15 highlighted with the G20 that we are taking their pavilion right now. The partnership between KAUST and NCVC showcase a successful story that can be implemented between entities throughout the kingdom, nationally and internationally. The opportunities for collaboration in genomic and ecological research is very important in restoration and greening in Saudi. As we are talking about this presentation, we are calling for action to global stakeholders to join this restoration effort in the kingdom. The future direction in the NPGP or the Native Plant Genome Project can be highlighted in the three stages. First, to expand the research to 300 native species that are listed in the Saudi Green Initiative. Next, we will be able to develop a genetic biodiversity index to monitor the ecosystem health. And then we will be able to inform policies for a larger scale restoration under Vision 2030. So we will be able at the end to create those policies and support it with the government. So in the end, before we finish with talking about those two projects in collaboration with KAUST, we cannot stop thanking the both teams from NCVC, that was led by Professor Ahmed Al-Ghamdi, and the teams in KAUST that were led by Professor Rod Wing and his four subgroups that are in KAUST. And we cannot stop thanking the postdocs, the research scientists, and we cannot forget the students. There are several graduate students that were involved in those projects, and some of them are Saudis, which supports our global initiative. And now we can move to Dr. Zhao's presentation.
Okay, thank you very much. My name is Zhao Xunyong from the Chinese Academy of Science of China, and I mainly focus on the dryland ecological research in eastern part of China and to combat desertification together with poverty relief in east. And the original landscape is look like this one, and I will follow the four content to give an introduction about desertification research and combating desertification in eastern part of China. And but now, 20 years later, it's about the 1990s. It's changing into this kind of landscape. Which I like to call it one photo and many systems in this kind of situation. But since middle of 1990s, the desertification trend is reversed to some extent, mainly beneficial from the national-wide campaign to against the desertification in China. And I will follow this. Is the map. As we can see, this desertification is the most severe situation in the world. And this kind of landscape, but now it's come back to China. Nearly one third of the land of the country is threatened by desert expansion and the desertification. And also the national project is actually, how to say that? Right along the national, one of the national projects is mainly along the blue circles and even to the western part of China. And in my research area, it's a transition area from the humid and subhumid areas, so it's much more fragile. It's also a transition area from tree forest to the grassland. That is why we come here. And I think it's very nice. This morning we have to talk about adaptation. That is very nice to have this photo. And China has set up monitoring stations over China. It's about 120 stations. We have the same measurement, equipment, and monitoring schedules to see how the ecosystem changes. We call it, and the climate change is quite clear in China, and it's almost much more like the rest of the world. That the most significant change is the land use. With the development of China economy, the cropland invaded from the eastern to westward. This causes a severe problem, but because the climate in the eastern part of China is much more adaptive to cropping. But due to development, the cropland reduced and also invaded to the eastern part of China. In the past 10 years, I have carried out the research to monitor the plant, soil, and water changes in these two blue lines to see the change. And also, one of my students to carry out the plant the relationship between plants CNP and the soil CNP. We try to stick up the quantitative relationship between the plant and soil in different degradation degrees. And also this one is we base it on the genetic series. We see the form of the local species are different. So we carry out the research to see the dominant species Artemisia, Halodon jaw, but this species has been divided into eight haplotypes to adapt to the shifting sand situation. But based on this kind of situation, we establish a permanent long-term experiment to see how this kind of change can be convergent together. And also we carry out the long-term research, use the facilities to monitor the changes of above and below ground ratios, and also the material changes and the water distribution from the precipitation from the center to the edge of bushes, to see this kind of change, to see how this kind of plants adapt to the dropped species, dropped stress. And then we can carry out the research based on the water consumption of individuals, populations, and even communities to compare these kind of species. The first one is Haloxila Caragata microphylla and Tamarix Hamosis, and different species, but the average precipitation is the line is not disappeared. The average precipitation is 300 to 550 millimeter. We can see most of the local species consumes water less than 300 to 550 millimeter, but some species much higher than that. With this kind of figure, we decided the proper tree plant density in the range of 220 to 370 per hectare, much lower than the current tree planting densities. With this kind of species selection and density, and then we get a good result. And this is a student dormitory, and based such kind of research, and the station, and in cooperation with other stations, we have adopted different sand restoration, how to say that, desertification restoration, and the project protection system from eastern part of China to middle part of China and to western part of China. This kind of made some successful stories. And for example, my station has been adopted by the UNEP and FAO as the successful cases for combating desertification and the poverty relief in 1990s and up to now. And we use this kind of local species and mechanical measures to fix the substance and encourage the invasion of different species, local species. And also this is a very successful cases. And at the same time, we select adaptive plants and livestock to carry out the poverty relief works. When we know desertification is driven by climate change and overuse of land resources, but who overuse land resource? The local people, and then we shift to support local people to improve, lift their life standard. We made also this progress, and we always develop new techniques to increase the income. That is why we made some achievement. But anyhow, we still faces challenge. For example, when the desertified land increased and decreased, but it's almost fluctuated. It's quite difficult due to the increase in population and livestock numbers. The consumers, as we can put these together, and it's quite difficult to us make progress. And also the reduction of water, about the reduction of the above ground and the below ground, and we faces severe problems of the degradation of natural plants, and it's quite difficult for us to revegetate. This kind of is a severe challenge. So let's come to this nearly to conclude my research, and we set up this kind of research framework from molecular element to ecosystems to carry out the research and monitor the changes of ecosystems based on a study in 1988. And this project we call it 100 project, and we have this kind of facilities to monitor the changes, climate, soil, water, vegetation, and including microorganisms. And then we come to this kind of figure. We can see quite clear. I took the first photo in 1989. We can see the student go to the school in sand dune area is quite harsh for the student. And now we can see it's quite clear the Mongolia students share their happy study life in the school. And also I suggest we can set up the long-term monitoring site across the Mongolia plateau to promote joint cooperation between Mongolia and other Asian countries to work together to fight desertification. At last, combating desertification must more like climbing the mountains. But fortunately, in 2022, I climbed up on the Himalaya mountains about 5,000 meters above sea level. We can see, we can conquer such kind of heat, and also we can conquer desertification if we work together. Thank you very much.
Okay, thank you, Dr. Zhao, about the presentation. It's really interesting. So as you conclude those presentation, we can continue to the panel discussion. Um, I do have an interesting question to Dr. Zhao. So in arid regions like northern China and Mongolia that you were describing earlier, what is the most surprising discovery that you actually came across to in restoration practices, and what might be similar approaches that will benefit from Saudi Arabia that we can actually use in our country? I don't know if you have.
Okay, thank you for your good question. And at the beginning, like most of the scientists have done in the world, we purely carry out the measures to fix the shifting sand dunes with different plants, local plants and introduced plants. But as my experience in the past 37 years, something like that, actually we found the, how to say that, the participant of the local. That's very important. Yeah. When we set up the stations, researchers, and show them how to use their cropland, how to use their grassland, protect their forest land. That is very important because the participation of the locals can encourage us and also protect their environment and educate their kids to continued effort. That's the first. And the second is the enhance the, how to say that, enhance the capacity building of application of research findings. That's very important. I think that's very unique, or that's very useful for different countries. And we can call that maybe a universal idea understanding. We can say understanding, yeah, because the local farmers and the farmers and the herdsmen, they are the key. They are playing key role in land use management. If improperly used, land degradation continued. If used properly with our support and cooperation, we can see not only international cooperation, community cooperation, and research scientists, cooperation between scientists and the locals are equally important. Thank you.
Yeah, I totally agree that we need to incorporate, people and farmers, yeah, to reach the same goal, and to combat desertification. Okay, so I'd go to Professor Ahmed Al Ghamdi. I have a question for you. What have been the most significant challenges in using native plants for large-scale restoration in Saudi Arabia, and how are these being addressed through the current projects?
In fact, maybe the major challenge with the native species is the lack of knowledge about how to plant these native species. We know, even indigenous people, they know a lot about the plants, but we don't have reports. We don't have database about our plants. That was one of the goals of this project, to update our databases for our local plants, to understand them in the field and start them also in the lab. So that's why also we are working with our partners KAUST now and to do other, I would say, side of the study in the field very soon, inshallah. So we'll be studying our plants in the field and in the lab in order to understand the whole, I would say, aspects of our plants, and then to think how to plant the right plant in the right place. This is really important. Even though we have 2,300 species all over the kingdom, but not all of them are applicable to be planted everywhere. We need to choose which is the right plant for the right place for the. So this is, inshallah, will be one of the outcomes of this study with our esteemed partners from KAUST.
Yeah, I totally agree. We need to understand plants and ecosystems so we can plant each species in the right environment. So, going to or actually Professor Rod, how do you envision cutting edge genetic tools to influence restoration efforts over the next decade, particularly in addressing the challenging posed by the climate change?
Is it working? Oh yeah.
Yes.
Okay. Can you repeat that? I'm
Sure.
sorry, I was looking at the microphone.
No worries. How do you have what's the vision of the cutting edge genetic tools influencing restoration efforts over the next decade? Although we know that we have the climate change, and how can we address that with those cutting edge tools?
Okay, that's a good question. I mean, my again, my view is, so we have the 300 plants now that have been selected for greening Saudi Arabia. So if we, our goal is to really characterize these at a very, very deep level. And like I was saying with like the Salvador persica, if we could really achieve this goal of combating desertification at the same and watering with salt water and producing fruit, that would be the ultimate. But that's and we can do this with the cutting edge tools. So each one of these plants has the potential for that. And then Mohammed Saleh, he's a pharmacist, and he's looking at possibly discovering new biomolecules, new substrates that could actually be used in medicine. And so some of these products that we're actually combating desertification with could be used to extract new medicines. So those are some things. I mean, there's also the idea of using gene editing, for example. So the date palm is a classic example. It drinks the numbers I have is it drinks about 170 liters of water a day. That's a lot of water, right? And my famous phrase is, it's hotter than hell, there's no water. So how do we grow crops in an environment like that? So one possibility is using gene editing to, let's say, make the plant so it takes half the time to mature, and it could be half the size. So by doing that, we can actually have a plant that maybe is much more efficient in food production as well as water consumption. So there's a lot of many things that we can try.
Yeah,
So,
I totally agree.
okay.
And also we have the station, the KAUST NCVC
Yes,
station.
our
so we
experiment
can probably-
station to utilize this. So
Sure.
we invite scientists from all over the world to come and try their new ideas. I mean, I liked I think your thing on planting density,
Yes.
and water usage. Those are things but those are long-term experiments. So you can't just do that in a in a couple of days.
Yes,
So.
I totally agree. Going to Dr. Nahad, I do have a question for you also. What are the challenging, since you were the one of the leaders of the NCVC KAUST project, what are the challenges and opportunities in genomics and metabolome and microbe root architecture research for restoration? How can we utilize those, and what's the challenges, that we can expand on it in the future?
Okay, thank you. That's a really good question. But I'm gonna talk mostly on the genomic point of view, and I will leave Magid and Mohammed to comment
Sure.
on the microbiome and the metabolome. The first challenge is that actually we lack the genetic information of the native species.
Yes.
So that's the first thing we want to do, is to build an infrastructure that we can build on. And then the applications of the genomic studies are, you cannot count like how many applications you can do. One thing is with this huge plantation efforts and the Saudi Green Initiative, you're going to need to plant billions of trees.
Yeah, that's
And one thing we can do with with the genomics is to actually confirm the identity, the genetic and the regional identity of these plants, to prevent the accidental propagation of non-native species.
Correct.
Or in like non-regional genotypes. The other thing is the genomic and genetics will help identify genes that are actually associated with the heat stress, with high temperature, with the low water, and then with biotic and abiotic stresses. So with that, I think it's very important to start building an infrastructure, and then we can build on. For example, our aim now is building is generating high quality reference genomes for the native species, and then next that's going to help us in doing population genetic studies, and that would help us assess the genetic diversity of these native classes across their range in Saudi Arabia.
Wow, that's a huge project, right?
Yeah, actually,
and
it's just-
interesting. Yeah. Okay, I think we can go to Dr. Majid. How again? What are the challenges and opportunities in the microbiome?
Okay.
Not the
So
metabolome. No, microbiome, and then we'll go to the
microbiome.
metabolome. Don't worry.
Okay, so I can see there's a bit of confusion about microbiomes
No, no,
and
no, no, no.
metabolomes.
I just pronounced it mistakenly.
Okay,
No.
so for microbiomes, challenges was the vast amount of data was being generated, because we were not expected that the root of desert plant are very rich
Yes.
and highly diverse with different microbes with different functions. Because we are extremely or very lucky to see that they have different kit, if I can say, like the Swiss Army knife. They have different tools for every positions. So we are always going for a soil. It's really poor, no nitrogen content. However, we find the plant is very healthy and green. And then we found out that you're already starting recruiting different microbes, which really are helping the plant for this nitrogen. So what I wanted to say is that challenge was more advantage for us.
Okay.
That we can how can we deal and how can we interpret all these data, and in the end of the day we can have a take home message, because the idea it is not only to profiling the microbial community, but how can we translate this microbial profile to an application can be used for the next step? Because now we know the genomics, we know the microbiome. So what next? How can we use this one? And this here what we are really trying to do now, finding this way of summarizing, building what we call it a syncom or synthetic community. We know exactly what we are missing as a function or what is our target. So our target is decreasing the water use or maximizing the water use efficiency is number one. Number two, would like to reduce the amount of fertilizer if we are going to add something in the beginning or chemical in the beginning. Second, would like to have a root architect are going very deep to stabilize the root system as well as to capture all this water. And surprising, the microbe can help do that because the microbes are secreting different compounds, and this compound can play as a plant hormone, which really enhancing the root system. So what is we are doing with this synthetic community now that we are assembling the function we are looking for. We have a wish list, if I can say.
Yes.
We would like to have a plant is a legume plant can fix nitrogen in the rhizobia, for example. So put a rhizobia. If there's non-legume plant, can we put some microbes which can help also fixing the non-air atmosphere?
The soil.
And this-
That's what actually what was my second question. So, can we, after understanding those dynamics of the microbiome, can we actually utilize it into harsh soil like environments, and we can actually plant them?
In fact, as I'm speaking with you, we are doing right now.
Okay.
Because what we learned from the Native Genome Project, number one, it was a huge amount of data that we can assemble all together with a way of like a survival kit.
Yes.
We can just provide what is the plant needs. And the idea is not only to seed the plant and leave it, we need to seed the plant
Enrich
and
it also.
survive and continue as well.
Yes.
And this is actually, I'm not really wanting to say that microbial do everything, but in fact, it is doing a big part. And the knowledge we learned from the genetics, we know exactly what we are targeting because we are not going to change the physiology of the anatomy of the plant, but the microbial can change and help for increasing more.
Yes.
And then I think this will be complemented with with metabolomics,
Yes.
and then I think Muhammad can cover this part from our side.
So, Dr. Muhammad, do you have any additional to?
Yeah, so one of the most challenges in metabolomics studies when you compare it to, for example, genomics or proteomics, is that the data are much more complex because every single plant produces some common metabolites, but every single plant produces an atlas of a specialized metabolites. And of course, one of the challenges in metabolomics is how to identify clearly the number of not common metabolites, but that are specialized metabolites that are unique for this. Of course, for this, you would need some public databases that allow you to identify these compounds. But from the other point of view, that most of these plants that are in KSA are underutilized in terms of application, I mean, and other application. So it's not that easy to find a database that you can compare a plant from USA, for example, to a plant that is coming because this has never been done in this very detail. And for this, we try to build a manual database for every single plant out of these 15 plants with the idea of providing an atlas of the specialized metabolites for every single plant. And by the end, we can have something like a network between the Saudi scientists that are working on this metabolomics study to get an idea how to identify these compounds again and to provide this database for the future generations who are planning to use this for a specific purpose and then utilize the information directly. Of course, from the other point of view, we screen some of these plants, something like the Salvadora persica, for example, for screening for some bioactive metabolites that can be utilized in either medicinal applications or agricultural applications, something like bioherbicide, biopesticide, or something like this. And from this point of view, after screening, we can go for targeted isolation of these pure compounds and then rely on NMR spectrometry or something like this to identify what are the targets that could be promising for utilizing it into production or providing nutritional value or medicinal application for these specific compounds.
That's great. I hope that we'll create the atlas and we'll have a database that we can continue on building on. Okay, I think we can conclude. Anyone wants to add any comment? Oh, sorry, I had a question for you, Dr. Asil. So, what are the strategies do you have? Do you believe are critical for ensuring that restoration projects achieve intangible ecological and social benefits? Do you want me to
The strategies?
Yes.
Basically, when we talk about restoration, in my opinion, it's most important to talk about sustainable restoration. Sustainable restoration will start with having a data-prone approach where we collect data to find areas with the greatest ecological importance, identifying those areas, and then continue on monitoring and re-evaluation, having evaluations throughout this specific region, then apply it to other regions. In this way, we'll have a future, like a greater results for that restoration of the areas that were selected. We cannot also forget what Professor Zhao have mentioned, the local importance, to make the locals and identify and have the knowledge of the importance of restoration to be able to maintain it, not only by the government or the people who are doing the restoration, but by the locals as well.
Yes, I totally agree that locals are really beneficial in sustainabilities, and educating them will actually help everyone. So I thank everyone who came here, for the speakers and the panelists. Together, let's continue to champion restoration through innovation and research and pave the way for sustainable future. Thank you very much. العفو يا